Well, welcome everyone. When thinking about what I talked about this year, I thought that really adapting agriculture to climate change was probably a good overarching issue to address in the work that we do on genetic improvement of crops. The key driver really is climate change. We are having to address the changing demands of consumers for food and all of those other issues that determine the objectives of our plant improvement programs, but really climate change really seems to be the overarching issue that's driving a lot of what we have to do in crop genetics today, and it's really an issue I'd like to explore from a number of different dimensions. I'd like to take us right back to the beginnings of agriculture, because I think the evidence is that agriculture was invented as a response to climate change. That at the time we had a very rapidly warming environment in that Fertile Crescent area, and this is wild barley that I photographed near the citadel in Aleppo, and it's growing on the walls of the citadel. This is before it was bombed recently, I think. This is what we believe was the first plant domesticated around 10,000 years ago. And this was the beginnings of agriculture, where human societies came together and were able to form settlements and so forth. This is at least a classical view of where agriculture came, and it was driven by the necessity to produce food in a drying environment where the other methods of food production were becoming less viable. We've looked recently, and I think I talked a bit about this last year, at a bit more about the history of this, and with the benefits of large-scale genome sequencing, we've looked at a lot of barley varieties and a very large number of accessions from around the world. And one of the big questions that arose from one of my PhD students working many years ago with the barley collection in Aleppo was that we found that the Tibetan barleys were much more closely related to the wild Tibetan barleys than they were to the wild barleys from the Middle East, where barley had first been domesticated. And this was part of what became a popular argument that maybe there was a secondary domestication of barley in Tibet. This was particularly popular in China, as you'd expect. There was a real interest in this, and it's great to think now we've actually finally resolved what happened by sequencing everything and really looking closely at the genomics of these species. And to cut to the answer, what we found was that barley really was just domesticated originally in the Fertile Crescent back 10,000 years ago, spread out from there. When it came east, it went through India 5,000 or 6,000 years ago, where agriculture was quite well developed, and moved up through Nepal into the Himalayas, going through a genetic bottleneck because of the cold temperatures. Only a small proportion of the population survived at those high temperatures. And then, after a period of cultivation there, escaped into the wild. So the wild barleys in Tibet, we believe, are escapes from the domesticates, rather than the other way around. And we can see that by analysing very carefully the genetic sequence of a number of the key loci that are involved in the mutations associated with domestication and then moving back into the wild. So it shows how we can actually unpick the history of agriculture by really looking at great detail at what actually happened. And that's been fitted in now with a lot of the archaeological evidence. And so more recently we've turned our attention back to Australia, because I've been working now with our local archaeologists and really revisiting the questions of agricultural origins in Australia. And there does seem to be a fairly substantial body of evidence for what we would possibly call, or probably call, agriculture in Australia a very long time ago. And it's just remotely possible that agriculture happened here first. We really don't know for sure. So we're looking at a whole lot of lines of evidence, mostly archaeological, but we're interested in starting to look at the plants themselves and how they may have been impacted in Australia by this very long period of human habitation here in Australia, where plants, animals and humans have interacted over an extraordinary period of time, probably more in the one environment than anywhere else in the world, and developed very sophisticated food production systems that involved all sorts of planting, harvesting, storage, transport and processing, particularly of grain. This area here in central Australia is what we think was a sort of grain belt. The climate was a little bit more favourable just even 1,000 years ago, and we think 3,000 to 5,000 years ago we've got a lot of archaeological sites. This one here particularly excites me, where we've got mining on a large scale of grinding stones. This is a large scale quarry used for grinding stone manufacture, for processing grain. This is about 3,000 to 5,000 years old. So there's a really long history of grain processing utilisation in the country. We're just starting to untease, and a lot of that is going to, I think, be able to be critically analysed by looking a lot more at the genomics of those very early those populations that exist in Australia. Interestingly, in this area, we think that in the Channel country where we're focusing, we think that there were two big industries a few thousand years ago: the grain trade and the narcotics trade, because narcotics was sourced from that area, and that was a source of wealth for people in these areas, trading agricultural commodities with other groups to the east, for example, that grew different types of crops. So there's a whole new area opening up of looking at agricultural, Australian agriculture, and I think what this will do is give us a whole lot more understanding of how we can work with agriculture in the Australian environment and discover a whole lot of things that have been lost. And part of this brings us back to some of the popular documents. I don't know how many of you have read any of these books, but I, many years ago, really interacted with Jared Diamond. He produced this book, Guns, Germs and Steel. He's at Davis, and he really tried to explain the history of humanity based on the interactions with agriculture and food production. And this was a very popular book, very widely read and sold, and really he posed the big dilemma of why the traditional view was there'd be no agriculture in Australia. And he couldn't explain it because all the evidence was against it. But we now have had this book, Dark Emu, which has become a bestseller in Australia, which has proposed the argument that we always had agriculture in Australia, and it's really a cultural thing that we didn't recognise it a couple of hundred years ago when we arrived as Europeans. So I think there's a whole lot of coinciding of the science with some of these really best-selling books now that have really captured the public imagination. And what I think the archaeologists are doing is racing to catch up with what these new perspectives give us about the long history, potentially, of agriculture in this country. One of the issues that's related that we're dealing with is how to come better to terms with all the intellectual property issues that are implied by all of this. So we're now trying to provide really global leadership here on the issues by taking a leadership role in the DivSeek initiative at the international level, and really trying to come up with guidelines for people that can really ensure that scientists can be compliant with things like the Nagoya Protocol. And this is not easy. The legal situation is incredibly complex, but we're trying to demystify that for people and trying to get local Australian legislation to actually harmonise with what we've agreed internationally on these issues. In terms of genetics to deal with climate change, we really are facing the challenge of moving targets. Much of our resources focused on really equipping plants to perform well in the field where they are now, the conventional sort of approach. What I think of as the second approach that we're seeing more of is to move agriculture to new environments, to shift to a different region. And I'll go through examples of that soon, where really the response is, we see the wines moving further south, we see all sorts of things happening in terms of where food is produced because we're trying to chase the climate. But the third option is to say, well, we're going to have to modify the environment. We can't actually produce agriculture out there in the environment. We have to actually look at various forms of protected cropping. And of course we can protect with a shade house, we can go into a glass house, or we can go completely indoors, completely control the environment. In horticulture we're seeing a significant proportion of production at the high end going quickly down that path. So we're going to see much more of our food produced here relative to here in the future. The speed of that is hard to predict, but it could happen very quickly. And for some crops we're seeing almost complete conversion to this type of production system. But for those of us trying to breed crops, of course, that completely shifts the goalposts. We no longer are trying to breed for variable climate, we're trying to breed for optimum performance in a perfect environment, or optimum performance in an environment that we can generate at low cost. So there's a whole lot of scenarios of deciding which environment we're going for and what type of genetics that we want. But this, for some crops, completely changes the paradigm, and we really are at risk with a lot of horticultural crops if we continue to work back here when the world is moving here very quickly. And a lot of the plant science we need to do this is really quite cutting edge, because we've never been really faced with what is the optimum environment for the plant and what is the optimum genetics for that optimum environment as the main objective. It's always been some sort of compromise about things in the environment. And so I think our key strategy remains turning to the broader genetic resources that we've got available, and particularly our unique opportunity here is the genetic resources that we've got available in Australia. And I want to focus largely as the example on rice, because rice is a really great example of what's possible with Australian genetic resources. And we're really looking very broadly at the question of how we can reinvent rice. We've now got a complete understanding of the rice genetic tree by really looking at the genomes of really the whole genus. There's just a few species that we're working on still, but basically we've put it all together. A great variety of food types, and really spread pretty much around the pan-tropical world. What we're able to do is really understand how these species have evolved. Now that we've got the detailed genomes, we can look at the various steps that have been involved in one species evolving into another, and how that's happened over a long period of time. Particularly focusing here in what we call the local area, which is from here to northern China and across to India, which is where the wild ancestors of Asian domesticated rice are found. And we've colour-coded the different genotype, different groups of rice that we detect across those environments, and you see the red Australian ones are quite different, although we've found some of the Australian material up as far as the southern Philippines. So rice is very much a native plant in Australia. This is a common rice that we see in North Queensland. This is in the Kimberley. You're travelling through the Kimberley, through the Boabs, and you look out, you see in the dry season any areas that collapse like that. It's usually the rice, because the rice lodges when it dries out and falls into big clumps in the field, so you can pick the rice from a large distance away because it's all collapsed, because you get these pure stands wherever it was flooded or there was a depression on the plain where rice will fill that space and it will collapse in the dry season. So just to show you some of the things we've got in Australia in rice, in terms of the options we've got to produce rice that better suits future climates. We've got this species, which is probably the closest species to the domestication clade that exists in the wild. And we call it Reefy pogon, but that's because it looks like Reefy pogon from Asia. But we now know for sure it's a different species. The DNA really shows it's completely different. So we're working with the herbarium to give this one a new name. So this will be a new, uniquely Australian species, widespread in Queensland across probably just into the Kimberley. This species, further out, this is the most distant of the primary gene pool of rice, Meridionalis. This has both perennials and annuals, so it's a really good system to understand the evolution and genetics of perenniality, because we can look at it within a single species, and we can look at it across environments. This is the one that we've had to chase in helicopters because it's on islands in Torres Strait. It's a much more exciting one than some of the others to find. Not sure, we haven't been able to find it, and I think in recent times, and it may not have persisted in the environment, so it may not be in the wild in Australia at the moment. We don't know where there's a wild population. Lots of collections 100 years ago. This one, very widespread and really unique, different lifestyles. We've got all sorts of lifestyles in these plants. This one survives the dry season as a rhizome, something that really offers new opportunities for rice that could grow in very different climates. So the others survive as either seed or they only persist in water as an annual. This one dries back to a rhizome and reshoots again in the wet season. A very different biology and somewhat more distant. This one doesn't quite make it into Queensland. The Queensland herbariums work very hard, but we can't find a plant this side of the border, unfortunately, so it's a New South Wales plant. This one, we had a wild rice workshop here, and the Illumina rep came in the next week. He brought in a sample from his backyard that was growing as a weed. So this is very much a local plant. And this is more distance. The genome is now being sequenced in the US. So we've got quite a range of genetic resources across northern and eastern Australia that allow us to think about quite new types of rices. We've identified particular targets for domestication. The key thing is shattering, the fact that the seeds fall off at maturity, and we know, of course, the genetics of that, so we can rapidly deal with that, and we've now got projects that will aim to use gene editing to really rapidly deal with these domestication traits so that we can rapidly domesticate new species of rice. I've just turned it off. Yes. And we've been working with collections, particularly from Townsville North right up to the tip of the Cape, and there we've got a lot of diversity in flowering time, a whole lot of genetics that's really important in rice we can find in the field, and so we can understand the genetics in real populations of these species. There's huge populations. In some areas in the north, it's just rice with them. That's the understory, and we can't see anything else except really quite pure stands of rice. In northern Australia, we have all sorts of interesting biology, including a whole lot of rice diseases that are unique to Australia. And this is one of the challenges of growing rice in the north. As rice has evolved independently in Australia from Asia, so have the rice diseases and pathogens. So we have our own strains of blast, we have our own strains of all of these diseases that are quite pathogenic on Asian rices. So when we try to grow Asian rice in North Queensland, the diseases really give it a hard time. We lose a lot of crops. The native rices, of course, have evolved and have resistance to that, so we've got an obvious path to producing resistant rice for the north. And the useful traits we've got big grains, colour, all sorts of interesting starch properties in the north. These big grains are interesting. This is an Australian wild rice, and this is an Asian rice. So you can see we've got the potential to get bigger grains by going into the wild, and some really interesting genetics there. We've already back-crossed with the Japanese, back-crossed that single gene for grain size, 25% more grain size into rice. Probably very attractive to consumers, but probably also going to contribute to yield. We haven't got data on that. Interested very much working with Bob Gilbert and others on the novel starch properties and the sensory evaluation with Heather. So we think that these rices are particularly attractive as new food products. So just to summarise what we know about rice evolution now, this is the Asian gene pool. This rice was domesticated in Asia here. These are the African wild rices, and this is the Australian material. So this clade here that has the domesticates in it, this taxon A, the rice we're going to give a name to hopefully soon from Northern Australia, is a sister to the whole clade. And then outside that is another clade which includes annuals and perennials that diverged in Australia about 3 million years ago. So this phylogeny says it's most likely that the clade starts in Australia, diverges quite a bit, and then finds its way in more recent times, just in the last million years, into Asia and Africa. So we have a clade that's got a fairly strong Australian origin at the base of domesticated rice. So in terms of many of the climate response things, one of the really great examples of how understanding the biology and the molecular biology of something can really help is our discovery of fragrance. When the rice genome was first sequenced, within weeks we had discovered the fragrance gene. I had a PhD student work from either end of the chromosome until, and because we had the complete sequence, we could find the gene very quickly. And we had no idea what the gene was, but we very quickly understood the chemistry because we discovered an aldehyde dehydrogenase. They catalyzed a reaction that effectively made the plant susceptible to stress. It actually knocks out this proline metabolism pathway, which is an immediate response of a plant to abiotic stress. So fragrant rices are quite sensitive to stress. That's why we get poor yields outside high quality production environments. They're extremely susceptible to salt. So what we've looked very widely as ways to get around that, and the answer is that we've looked in pandanus. Pandanus grows at the coast, it's tolerant of salt, but you can also get fragrance. So in fragrant pandanus, we found a different pathway that's got a bypass for the steps that are blocked in rice. And so we now have a single gene from pandanus. We think that in rice will allow us to produce fragrant rice that's high yielding, because we'll be able to restore the ability of the plant to respond to stress. So I think this is a good example of how actually understanding the molecular basis of something leads to some really new opportunities to produce a climate resilient crop by actually understanding the molecular biochemical basis of the problem that you're dealing with. So the history of rice in Australia is it's been grown everywhere, but the main successful rice production, of course, has been in New South Wales, where we've been producing the order of 1 million tonnes. Australia's been a major exporter, 90% of our rice is exported, but it's all been produced in the Murray-Darling. That water is running out. There's really no capacity to produce rice in the future in southern Australia because the water is not there. The perennial crops are demanding the water. In the north, we've got natural rainfall, and we can grow rice under that natural rainfall. We can grow 2 crops a year. We may even be able to grow 3 crops a year because of the temperatures. A really great production environment, and what we do have to do though is reinvent rice. We have to come up with varieties that will actually perform in the north, that will cope with the diseases that are there. And so the real opportunity here is our response to climate change, the lack of water in the Murray-Darling, is to move to natural rainfall. It's a really good example of how an industry is going to probably completely relocate in response to climate. But in doing so, need to use a lot of genetics to get the right varieties to perform in that new environment. So at the moment we're exploring all the options. We're working with Indigenous people on wild harvest options, domesticating, working with domesticated Asian rice by adding in things like disease resistance from the wild, but also working the other way with wild rices and trying to domesticate those, but also having the long-term objective now that we've worked out with industry to develop a uniquely Northern Australian type of rice. You know, you've got basmati rice and those types of rices. We want something that's of similar stature, that's uniquely Australian, picking up natural attributes, but able to be identified with Northern Australia. So while I'm still on rice, I just say that with all the genomics we're doing in plants, we've really come a long way with things like the chloroplast. We now can get the complete chloroplast sequence very easily. We can annotate all the genes, so we can get a complete set of all the genes in the chloroplast with every sample we look at as a byproduct of the other genetics that we're doing. What this has led us to is some very significant discoveries about rice just recently that show us that in fact there are 2 types of chloroplasts in rice that are functionally different. And this is not something that we expected. So we've got these 2 big families of rices looking at 3,000 rice varieties. We see that they break into 2 groups, and they're 2 functionally different chloroplasts, where we have genes that encode quite different proteins in the 2, but there's only 2 types of chloroplasts, and they don't correspond to the type of rice at all. So you've got japonica and indica rices in both groups. It's unrelated to the variety of the rice. This is something that has separately been selected. So in plant breeding and the history of rice, the chloroplast and nuclear genomes have taken separate evolutionary pathways. But what it shows us now is that we've got what I think are functionally different chloroplasts. And now that we know they're functionally different, we can actually work out what the phenotypic advantage is of that. And we've found differences in the photosystem genes that suggest that some of these rices will have different responses to different light regimes. So there's a slight difference, I think, in adaptation of these to different degrees of shade, and that's what we've seen in other riser species, that we can potentially change the optimal light requirement by changing the choice of chloroplast. And this is quite a new discovery because no one had looked before, because they didn't have the tools to look. But the fact there's only two types, but they're completely different. They've got lots of differences. They're like evolved a long way apart. Quite a remarkable hidden aspect of the rice gene pool. So to move on from rice and just cover some other species, we're trying to do with sorghum everything we've done with rice. So we've got the John Genome Institute joining us to sequence the whole genus, all of the species, and of course they're mainly Australian, mainly Queensland species. Sorghum is domesticated in Africa, but very much a Queensland thing. And so we're now looking at not only all the species, but the variation within them. We've got a very large effort to do very high quality reference genome sequences of all of the species from the wild, and to really understand the evolution and genetics of the sorghum at the genus level. One of the things that's bringing us to understanding in a project we've got with a number of other groups is to understand cyanogenesis in plants. Almonds are bitter. Sorghum under stress produces cyanide in the leaves, which is toxic for stock. Macadamias are mostly bitter because of cyanide, and we're really trying to understand the biological significance of that. The remarkable finding in looking at these wild species is that cyanogenesis is something that's in domesticated sorghum but not in the wild sorghums. So it's a trait we've actually selected for in domestication. There must be some advantage in the domesticated situation of having cyanide in the leaves under stress, whether it's to do with nitrogen cycling or actual stress tolerance, or pest resistance that's required in an agricultural context that's not required in the wild. But for whatever reason, we see in a number of plants now evidence that, bizarrely, it's a product of human selection, unconscious probably, human selection. Also, looking at wheat in this context, and there's a lot of, I guess our biggest sort of discovery there is that there's pathways of photosynthesis in the seed that seem to explain the performance of wheat under stress, particularly under high temperature stresses, that have really still been poorly understood. And that there's lots of wild relatives in Australia that we think have got really interesting attributes that we're just starting to look at. What we've found in wheat is that there's enormous diversity in the domesticated gene pool in tolerance to heat stress, way outside that the plant breeders are using. So this variety here, remarkably, is a Thai variety. So this gives us an idea that Thai varieties of wheat are pretty unusual. And these local varieties here, when we heat stress them, we get quality challenges, we get yield losses. The same heat stress on this actually results in faster growth rate. So we've got, so we've done some very interesting experiments with Canadian wheat. 37 degrees for a long enough time kills them. The local varieties suffer badly, all of them, but there are varieties that actually grow faster. And we're not using that great diversity of heat stress tolerance because I think people just haven't been looking. They haven't been looking with the right molecular tools to discover it. Because phenotypically, they may have discovered these extremes. Coffee for new climates, really important. I think lots of talk about what climate change is doing to the quality of coffee and production of coffee worldwide. I was in the US last year, I think, and I had the ABC chasing me because someone at Kew had announced that all the species of coffee in the wild were going to become extinct because of climate change, and people were really concerned. And it really, I think it's true that people that are not concerned about climate change are concerned about coffee. And so this is the way to get public awareness about the issue. You don't care about climate change. Don't you like coffee? This is really, and from the feedback I got from the ABC, this was one of the most popular stories I've ever had, that climate's going to affect the quality of your coffee. But in reality, we do have some wild coffees. We've looked at the whole group, and many of them are challenged because the habitats in which coffees occur are all threatened, particularly in Madagascar both West and East Africa and into Northern Australia. But what we've really found by looking again at the genome, we've got the genomics, we've got the transcripts, we've now understood that really this key experiment, which is just about to be published, shows that the coffee from the bottom of the bush is better than the coffee at the top of the bush, and that's because it's more slowly maturing. We look at gene expression, everything happens more slowly at the bottom, and this explains, I think, why the high altitude coffees are so good, because what we do with slow maturing, it's a bit like fruit. If it matures slowly, you end up with a higher content of sugar, higher content of caffeine. If you mature it too quickly, you end up with a less desirable composition, and you don't have that elite flavour. So the way to make good coffee is to grow it slowly, is to have that long, extended maturation period and we can see it in the gene expression, we can see it in the composition of the berries, and particularly the sugar content is important. There's no sugar after you roast it, but it's the breakdown of the sugar that produces a lot of the flavour compounds when you roast coffee. So if you start with high sugar, you end up with a much better coffee bean. And so a lot of how we go to deal with producing coffee in new environments can come, I think, from understanding what's driving that chemical composition. Well, I think we've got great opportunities with the pulses, because Queensland is just full of them. We've got so many wild pulses that are the key relatives of the major crops: soybean, mung bean, pigeon pea. This is where all the exciting genetics is in Queensland. It's nowhere else in the world, and where no one's really working on it. The work we've done with pigeon pea just illustrates, I think, particularly to me, that we've got the ability to capture genes that will really make a big difference to these things becoming crops, not just in Queensland but elsewhere. I wanted to talk a little bit about horticulture, and one of the big programs that we've got is a horticultural genomics program where we're looking at the genomes of these 5 groups of plants. We're trying to do detailed genomics here. These represent about 80% of the value of horticultural tree crop production in Australia, so that's why we can focus on these groups and make a big difference in Australian horticulture. And one of the associated projects that's continuing is to try and use this knowledge to sustainably intensify production of these, and particularly in response to the climatic challenges that we've got. So this is producing much more dense plantings of these trees that are much higher productivity than the current systems that we've got. Macadamia is the one that's probably worth saying most about. We're standing here in the centre of the natural distribution of the species. We're working with the genomics of these in great detail to try and understand what we can do with them. There's great interest in growing these in different climates, so can we really grow these in North Queensland? We're still not sure just how well we can grow them, but how well they'll do in North Queensland, where there's enormous interest in growing this as a crop. The species that we've done most on recently is Macadamia jansenii. This is a rare plant, originally only 30 plants known from the wild, recently discovered. We're now up to nearly 100 plants, I think, known in the wild. We have DNA from every known plant of the species. It's a great system to study because it's complete. We don't have any uncertainty. There's no plants out there that we don't have. We have every allele of every gene, and we will have the sequence of every allele of every gene very soon. We've done chromosome level assemblies now. We've got the chromosome size with this. It's going to be a really interesting example of a rare species where we can look at the issues of genetic bottlenecks in low diversity situations. It's a really important source of new traits because it's a small tree, and a lot of the small tree attributes are what we want to use in domesticated macadamia. It's also a basal eudicot, the angiosperm phylogenists here. That's really puts it as, this is now probably one of the best genome sequences we've got for a plant at the base of the eudicots. And this is at the point where we're seeing the evolution of the number of plant parts, the distinction of the sepals and the petals, if you follow that sort of differentiation. So we've made this a model system for testing sequencing and assembly of plant genomes with the UQ Genome Innovation Hub. So we've sequenced this with every available platform. We compare our ability to assemble those sequences, and we've also are using that obviously to go further and work in complete analysis of the system. So I think this sort of approach for key species is really going to be important. We're doing the same sort of thing with Duboisia. Here we're trying to increase the alkaloid content. This is a plant predominantly grown at the moment in Kingaroy, but we want to grow it in other environments. How do we take this into a broader range of production environments outside the Kingaroy area where it's native, but what we're trying to do is dramatically improve its productivity. We're working with jojoba, with our colleagues from Saudi Arabia. This is a remarkable plant in terms of its drought tolerance, so we're looking at gene expression in response to drought, looking at the genome in a lot of detail to understand the traits in this species that give it this incredible ability to grow in the desert. And of course, I think we're continuing to look for sites where we can look at wild populations growing across steep environmental gradients, because that allows us to look at how plants in wild populations are adapting to sharp climatic differences. So here in this case, we looked at the wild barleys on this is northern hemisphere, the north-facing wet slope compared to the south-facing dry slope. And we're able to look at what genes are different, what alleles are different in the plants that grow in those different microenvironments. We've similarly done the same thing with a very distant relative of rice, in the rice tribe at least, growing from Melbourne up over the Alps and back down to the ocean again. And by looking at plants that grow in different climates from the same species, we can really look at how those plants are adapting. We're taking that further with a major program now that's extending our work on the rainforest species to look at the genomes of many of the trees in the rainforests from sort of southern New South Wales up into Asia, and trying to look at the evolutionary relationships, how plants are evolving in that very dynamic environment of the rainforests. So I've been talking about adaptation, but I wanted to say a little bit about mitigation of the role of agriculture and plant genetics in mitigation of climate change. We're running a series of conferences. The last one we had in Budapest back last year. We're just planning the next one in Germany next year, where we're moving from a focus where we've had in our agriculture and climate change conferences on adaptation to how agriculture can actually help to mitigate, how we can actually use agriculture to arrest climate change. And of course, this is the sort of thing we're talking about, using plants to replace fossil fuel as a source of renewable carbon, rather than using oil, for example. Can we use plant biomass? Increasingly, that focus for us is not on where we were 10 or 20 years ago producing biofuels, but rather very much more on producing, to stop doing that, producing biomaterials, plastics, fabrics, etc. More than half the oil in the world is not used in transport. It's used to make feedstocks for petrochemicals, for those sorts of products. So it's the carpets and it's the plastics that can have carbon that's coming from plants rather than from fossil sources. It's one of the key areas in which we can make a contribution to actually mitigating climate change from agriculture. And so that's really about understanding the chemistry, particularly of the cell wall, the chemistry of lignin, and understanding the genetics of how we manipulate those things. And we're really focused on, in a number of systems, doing that. Sugarcane is our primary example. We've now got some sort of a first pass, a genome for that, but we're moving towards having, for this plant, one of the 20 top crops in the world, the only one for which we don't have a genome sequence now, and that's because it's so incredibly complex, but we're going to beat it. I think by the end of 2020 we'll have a very high quality genome, because the technology's caught up at last with even something like this. The other type of plant biomass that's been a real focus and is important in Australia are the eucalypts. The eucalypts are, I think, the other plant that at a world scale can produce the sort of productivity that sugarcane can, that 100 tonnes per hectare of dry matter per annum, in not necessarily even the best environments. That makes these amongst the most attractive options globally for that, and we've got unique access to the material here. We're working with trying to understand the genetics of plant architecture in some of the local species, and these two species here we're going to focus on to try and understand plant size and its genetic control. We're about to publish, finally, the Corymbia genome. That's the the spotted gum genome, and this has evolved, separated from the eucalypts, the corymbias, from the eucalyptus. These are both eucalypts. So this is the flooded gum locally and the spotted gum, both local plants. They diverged more than 50 million years ago, incredibly long time ago, from our good whole genome analysis. But what's remarkable is that in the Australian environment they've undergone parallel evolution. What we've seen in that time is an expansion of many gene families by the same mechanism as gene duplication. For about 30% of the genes, within 3 genes we find another gene that's similar, that represents a duplicate. And this has been a mechanism that the plants have used to respond to upregulating the function of things. And the two main functions we see are terpene synthesis and abiotic stress tolerance. It's the production of the oils. And what's remarkable is we can see it's happened independently, but it's happened in both lineages. So they've both responded to the climate in the same way, and they've both used the same sort of mechanism, gene duplication, and it's been in the same types of genes, not the same genes, but in the same groups of genes. So quite a story of parallel evolution in these groups. And these groups, because they've got small genomes, are really attractive to work with, to study a lot of basic plant biology. So really, in addition to looking at relatives of existing crops, I want to emphasise that we should be thinking about domesticating completely new plants to cope with climate change. If it's a grass, it could be a cereal. So that's the way we've got to think, and there's many options. So there's both crop wild relatives and completely new crops that we can use in expanding the gene pool. One of the initiatives that we're trying to align with is the Genomics of Australian Flora project. This is an ambitious goal to sequence all of the Australian species. We've got 25,000 species of plants, so that's not going to happen tomorrow. But what we're starting from is a top-down approach. We had a national competition, and unfortunately we didn't win. New South Wales did, and so the Waratah tilapia is the key target for sequencing. But very kindly they asked us to join the project to help them to do it, so we've sort of won anyway, I suppose. This will, after the pilot projects, work out to look for an example of every genus. So we're going to try then in the next phase to sequence an example of every genus of Australian plants, and then go down to species level. Quite an ambitious but a coordinated project through Bioplatforms Australia. And partly because of that, Bioplatforms Australia is supporting us. We're interested in agricultural things. We're going to get to sequence all the Australian citrus, the 7 species of Australian citrus, because Bioplatforms wants to fund Australian species, and because they're agricultural, we get the funding to work on them. So by working across all of flowering plant diversity, I think we're able to mine it for useful genes for both species and genes of interest. And what really is continuing to drive the advances is the remarkable advances in genomics technology. The quality of the genomes that we're now seeing, we're now having, are so much better than anything that's in the literature. The technology has moved so far in the last year, and I know I say this every year, but it really has moved so far in the last year that the things that are appearing in the journal pages today are things we would think that's probably not publishable. It really is an incredibly dramatic improvement in the quality and cohesiveness of the genomes we're putting together. And what it's giving us is an incredible list of things we can edit with gene editing to really make the crops of the future that are what people want and that actually adapt to climate. So TropAg, of course, we're running again in 2021. Associated with that, we're going to have the Harlan 4 Symposium. Harlan 3 was in Montpellier last year. This is about domestication of plants and animals and use of biodiversity in agriculture. So this will be an associated conference. We've attracted to Brisbane. The first symposium was held in the Fertile Crescent, the second one in Davis, the third one in Montpellier, where they focused on the origins of grapes and things like that. We're going to try and focus here on some of the Australian plants, hopefully on indigenous agriculture, and some of those emerging areas of research. So I'll just end by finish by thanking some of the collaborators. I think a lot of what we're doing in genomics is really only enabled by having a lot of collaborators. The key ones for us now are JGI. We're doing a number of really major projects with JGI, really partly in with support from the Joint Bioenergy Institute and also the Department of Energy, because they want to see the sorghums and the sugarcanes of the world sequence because of their importance as global energy crops. Our work with King Faisal University, the Genomics Innovation Hub, which is really driving the potential of UQ to contribute really fundamental advances in the technology here. And I think we're going to do great things in the Global Bioeconomy Alliance. We're just starting, of course, our ARC Centre of Excellence in Plant Success in Nature and Agriculture, and that's 7 years of research from July, so there's going to be a lot more work in this area. I have got a couple of minutes, so I just want to talk about a couple of personal examples. This one is a eucalypt that was bred by my father, and it's an F1 interspecific hybrid that really illustrates, I think, the ability to breed plants that are really widely adapted. So what he did here is he spent about 20 years searching for parents, and then he did this cross in about 1990. And this is a cross between a plant that grows on the Great Australian Bight, where you look into the Southern Ocean, and a plant that grows near Darwin. Two different species. And if you grow the mother here in Brisbane, it doesn't cope with the cold in winter, really struggles. You grow the father here, it dies in the summer because it can't cope with the fungi when it rains, when it's warm, because it's adapted to a Mediterranean climate. The hybrid here grows everywhere and has really been very successful over the whole country. So I think we've got the potential to think about quite new examples. I haven't talked about this one before because it was a commercial product, so I was actually potentially earning money from it, but it's now out of PBR, so I don't make any money out of this, so I can talk more openly about it. But it's a good example of that sort of thing. And the other thing I thought was particularly current is fire. Climate, of course, I think is certainly associated with those that elevated temperatures that we've seen, the very dry environment that's associated with those extreme fire events that we had this year. But managing our biodiversity in the context of fire is a really challenging issue, and this species is the one that particularly personal for me. This is a plant I actually did a biology project on when I was at high school, and I struggled because it grows on the top of Mount Tibrogargan. I know you know Mount Tibrogargan, one of the Glasshouse Mountains. It's a small population. There's probably only about 50 plants in the whole population. And I couldn't work out what it was, and that was because it was an undescribed species. And I found out only like 20 years later when someone described it that was why I couldn't actually key it out as a student. I thought I was a bad student, but it was just that it was a new species. So this one really illustrates the challenge, because in recent years we've seen fires go up over the top of Tibrogargan, people lighting fires all the time. They burn up the back of the mountain across the top, and the population gets burned. So this species probably needs fire, so we have to have the fires, but it also, the adults are killed by fire. So we need a period between the fires that allows the plants to grow and form seeds again. If that doesn't happen, then we deplete the seed bank, and we do that 2 or 3 years in a row and the species is extinct. And so we're seeing, and I think that's the problem I think we've got in a lot of the alpine areas, where many of the species need fire, but they need quite infrequent fire, because frequent fire will actually completely abolish the population. But it's only a problem for species that are narrowly distributed, but we've got a lot of those, and a lot of those are important, including a lot of the crop wild relatives. So what we really need is a very major investment in fire in the landscape. We need to have a lot of people lighting fires every day and putting them out, burning really tiny areas. We can't burn whole fields at a time. It doesn't actually, is incompatible with biodiversity conservation. So I don't know how we're going to resource that, but it's a real challenge for climate in the future. So, and I'll finish with Another local plant that I think is important for the future is Cissus antarctica. This is a potential rootstock for grapes in much more tropical environments. It's a group that's not probably, well, again, Cissus is the genus, but the analysis I've done with Maurizio Rosetto suggests that this is actually a genus much closer to Vitis. It's actually very close to grape and is, of course, adapted to the rainforest. So very different range of climatic environments to what we would see in the grapes that we grow that suffer from sort of mildews and things in this environment. Here's a really good example. So I think I'll finish there. Thank you.